You want to go through those steps again of for the ribosome? All right. Translation is converting a messenger RNA into a protein. This happens on the ribosome. Okay? Bacteria have two components. Everybody knows two components of the ribosomes. Bacteria is a 30S and 50S, 70S total. For the process to take place, you need to, it first comes together with what we call an initiation complex. All right, and that involves, obviously, think about it, the things that you think would need to be in that recipe for it to work. A messenger RNA obviously has to be there. And then it starts with the 30th subunit. And then you need that first tRNA that's loaded with methionine. In the case of bacteria, it's formal methionine. You get that first start codon. So all of those things need to come together with a bunch of initiation factors. So they all come together into what we call the initiation complex. And once they're all together, the initiation factors can go away. But that first loaded tRNA with formal methionine is at the P site, which is the donor site. And that makes sense. Okay, so right under here you have the start codon, meaning with the anticodon of this tRNA from methionine. And remember you have three sites. You have the P site, I just said this, the P site is the donor site. You have an exit site, and you have an A site. And we call A for acceptor. Look at that as the acceptor site, where it's going to accept the amino acids as they get transferred over with each subsequent, as that tape of RNA moves through the ribosome, shifting over. They're going to add new amino acids over to the acceptor site. Okay, so once all this is in place, the 30S initiation complex comes together, then the 50S subunit can come in. That tRNA loads up right there in that P site. This formal methionine is the first one. So that's how it gets started. Now, how it elongates and turns into an actual protein, an actual chain of amino acids. It's just that it rotates through these three sites. So you start with here where you have a tRNA that in this case has a couple of amino acids on it. We've been going through the process for a while. It kind of started midway in this video. But you can imagine, to help make it a little bit more clear, that very bottom amino acid should be a green ball. That helps you understand. It should be that all right, so that's at the P site, the donor site. So a new tRNA comes in here to the acceptor site. All right, and an anticodon to this 3-nucleotide codon. All right, for whatever amino acid that is. It comes into that acceptor site, and then the amino acid chain that's growing there at the donor site gets shifted over. They're called transpeptidase reaction. It's shifted over. Those amino acids get shifted over to the acceptor site. So every time a new amino acid comes into that acceptor site, its amino acid is going to be at the bottom of the line. So these two amino acids get shifted over there. So now this tRNA sitting in the acceptor site is empty. Okay, right there. And now the amino acid, that tRNA in the donor site is sitting empty because it's empty. It donated these two amino acids over to the acceptor site. And so now in the acceptor site, it has a 3 amino acid chain. All right. And then once that happens, everybody shifts to the left. The empty tRNA is going to go here to the exit site. That 3-chain amino acid chain is going to go here to the donor site. And now the acceptor site sits empty, and it can take a new loaded tRNA for the next codon, anticodon set along. And then the whole thing happens again. And then those 3 get shifted back over. I said something wrong a minute ago. Did anyone pick up on it? About the green ball. Right. It would be on the top. Good. I was wrong. The second one?
Yeah.
It didn't. So if you look in your book, you'll see that these are two completely separate figures. I just put them together to try to teach it in a somewhat fluid way. But it would have gotten really good questions, and that's why people get hung up here. When I learned microbiology and genetics, my professor made us do like this whole interactive play up on the stage in this auditorium. I went to a huge school. And how do we do that with you guys? Because it really makes people get it. So what happened is, this is just showing your initiation complex getting started. So if you imagine that this green ball up here, that gray amino acid is actually that hormone methionine green ball, then you think, then just one step happened in between there, in between them creating this figure and this figure. So this is elongation in process. This is initiation. So peek in your book and it'll help you understand that these are two totally separate figures. All right, anyone else? Okay, hopefully that helped a little bit, and it'll soak in, and we'll go over it again. Okay, so today's lecture, not any easier. It's not very long, but it's pretty thick. So we'll go through it nice and slow. We'll take our time. Those of you that are comfortable with it, maybe can help out. We're going to go through regulation of cellular processes. And particularly this applies to all organisms in genetics, but it's particularly going to relate to microbial organisms here today, bacteria in particular. And we're going to go through things like the lac operon. Everyone, who's familiar with the lac operon? All right, a couple of you are raising your hands. And it's if you haven't ever seen it before, it's not immediately intuitive. I think I probably didn't really grasp the lac operon until I learned it the second or third time. All right, so what we're really talking about though is regulation of gene expression. And we can think of a whole lot of reasons why a cell would want to regulate their gene expression. Who can remember back to a couple of lectures before that first test? I talked about enzyme feedbacks, and I talked about feedback regulation in enzymes. And maybe it kind of sat out there and you were wondering why I was teaching you that, and I said at the time, you'll learn in a couple of weeks, and here it is. It's because cells are going to have a lot of reasons to regulate their genes to conserve on energy. So if they have, say, a gene for a catabolic process, maybe one of the enzymes in glycolysis, or an enzyme, say, to break down the sugar lactose, which really has been a lot of fun today. But if that sugar's not available, then why would they bother to make those, transcribe and translate those genes? It'd be expensive, energetically expensive to produce a product that you don't even need. Likewise, for an anabolic process, if I'm trying to make something, I want to make the amino acid tryptophan, or I want to make peptidoglycan, or I want to make something, an anabolic biosynthetic process. Why would I bother to make all the enzymes necessary, all the machinery necessary to make that product if I already have tons of product? Why would I bother to turn on the factory, bring all the employees to work to make cars if I already have a billion surplus cars on my lot? So that's why we have gene regulation. And what we're going to talk about for most of this lecture is regulation of gene expression at the gene level. We're going to talk about operons. And operons are there specifically to regulate genes that need regulation. Contrast that with housekeeping genes that are what we call constitutive. Housekeeping genes are always on. Basic fundamental processes. All right, and then at the end we're going to wrap up with a couple of global regulators. I introduced you to the idea of a sigma factor. We're going to talk about some sigma factors, and then we're going to talk about a very special bacterial process called quorum sensing, which is actually a pretty cool thing. All right. So gene regulation. You can have inducible genes, and you can have repressive genes. And you can have constitutive genes. Okay? Constitutive genes, I just told you about housekeeping genes, basic metabolic functions, things that don't necessarily need to be turned on or turned off. So, based on what I just said, let me see how closely you guys are following me. Inducible genes. What would be an example of an inducible gene? Compare it what I just said. I just want to see if you're following me. example of a catabolic reaction? Glycolysis. Huh? Yeah, so wait, what are you saying? I'm not hearing you.
The enzyme that breaks the amino acid.
Sure, okay, yeah. Okay. So an enzyme that would break an amino acid, an enzyme that would allow you to digest proteins, right? Maybe that would be a catabolic reaction. But if you don't have any proteins in your cell at the time, there's no proteins in the environment to degrade, there would be no reason to induce that gene. You guys gotta stick with me a little closer, we're gonna go off the cliff in about 4 slides. Right? There would be no reason to turn a gene on if you don't have the product that you would like to degrade. That is an inducible gene. Repressible genes, on the other hand, are for biosynthetic pathways. Going back to that enzyme feedback inhibition. Alright? So, if I have plenty of a product, if I have plenty of tryptophan, just an amino acid that you might want to make. If I have plenty of peptidoglycan and I'm not growing, I'm not trying to grow, then I might have positive feedback where I would turn off, or even consider it more of a negative feedback, but turn off a repressible gene. I will repress that gene because I don't need to make any more peptidoglycan. Alright? So a biosynthetic pathway. And I said constitutive genes are just housekeeping, making ribosomes, things like that. Alright, so let's go through an introducible gene. Lactose. Not everybody can degrade lactose. Many bacteria can. If you recall, lactose is just a sugar. It's a polymer of glucose and galactose. Okay. Lactose is glucose and galactose. Close enough. Alright. And there's a series of enzymes that help break down that sugar. Alright. And something that's going to become important before this lecture is over is if I'm a cell and I'm trying to metabolize something, it's going to be far more metabolically beneficial to metabolize glucose than lactose. Just tell me why. The answer is obvious as you think. Why? Exactly. It's a monomer versus a polymer. Okay? Glucose is easier to degrade than lactose. And you can't necessarily degrade lactose because you need a special enzyme to break up the glucose and galactose. Alright. So, if I'm a cell and lactose is readily available out in the environment, then I would like to induce the gene. So this is an inducible gene. It's a catabolic process, breaking up lactose into glucose and galactose. And so you would want to induce that gene. Just for example. Okay, let's contrast that with a repressible gene. So repressible genes are genes that function in a biosynthetic capacity. For instance, production of tryptophan. Not all cells are going to be able to take tryptophan, that whole amino acid, in its whole entire form. Cells are going to actually synthesize that amino acid. Certainly bacterial cells. We're going to have the capacity to do that. And so, in that case, this is a repressed gene where, when, in this case, there's no tryptophan present, we need to make tryptophan. I'm a car manufacturing company. I need to make cars. I don't have any cars on my stock lot. I need to make cars. I need to turn on all the machines to make the cars. In this case, there's no tryptophan present, so the repressor would be inactive and transcription can occur. Alright? If tryptophan were present, we're going to see this again in a couple of slides. If tryptophan were present, then it would be bound to the repressor to make it active. That repressor would be bound to an operator site, and it would get in the way so that RNA can't bind. It would repress that gene. Hopefully the ideas of being inducer and being repressor are intuitive. Okay, so let's just go over it. I have a lot of text in here, but hopefully it helps. Control of transcription, induction and repression occur because of the activity of regulatory proteins and DNA binding domains. So that's the mechanism through which this works. And another word for this is an operon. Operons can repressors and inducers such that that gene can be written. And they can be either unrestricted.
Sorry.
Okay, so let's go through negative control first. So when a gene is under negative control, in the simplest sense, it means that something is bound to it and that is turned off. Okay, so whenever a repressor is bound, and we're gonna see examples of this clearly, pictures from your book. When a repressor is bound, it turns it off. That is negative control. So in this case, we have an inducible gene. So for this gene that has been induced under negative control, it's regulated by negative control, you have to have had something bound there to pull that repressor off. But we don't. This is currently under negative control. The repressor is bound to the operator, getting in the way of RNA polymerase binding to that gene and producing a message. Alright, and the exact opposite of saying this is an inducible gene. If the inducer wants to induce transcription to occur in a negatively controlled gene, then the inducer will bind to the repressor, pull it off of the operator, allow RNA polymerase to bind, and a messenger to be transcribed. Alright, that's with respect to an inducible gene under negative control. Alright, a repressible gene does the exact opposite. Okay, it's under negative control, when it's bound and nothing happens. Alright, so here you have a repressor protein bound to the operator site, and it has to have some sort of a co-repressor. Alright, if it has a co-repressor, it's bound and no transcription. Alright, but it's not under that negative control when there's no co-repressor. The repressor is unbound from the operator, the RNA polymerase combined, and you get a messenger. So that's negative control. Negative control means when something is bound, the repressor, then no treatment. Positive control means the opposite. So binding of a regulatory protein, in this case we call it an activator. And I'll give you a good example of this in just a little bit. And that's going to bind to the regulatory region of DNA and then transcription will occur. So under positive regulation, you are required to have an activator there for transcription to occur. Which is the exact opposite of negative regulation where if you have something bound, okay, positive control of an inducible gene. In this case, you have no inducer attached to your activator, so it's not bound, no transcription occurs. It's the exact opposite of negative. Alright? In this case, you have the inducer bound to the activator, which makes it bind up to the activator site. RNA polymerase binds, you get a message, transcription occurs. Alright, you can still have positive control of a repressible gene, and it's just the opposite. In this case, the activator protein is bound up to that activator site, and that's what caused, in the inducible gene, that's what causes, in the repressible gene, that's what causes the RNA polymerase to bind and the messenger to be made. Alright, but under positive control, you can inhibit that activator, pull it off, so when it's not bound, because it's positive control, then you get no transcription because it's not bound. Are you guys comfortable with this dichotomy? Hopefully I'm going slow. Hopefully some of you are bored. Okay, so let's think about why we care about all this. And then we'll get into the exact details of the offense. The reason you care about this is the economics of the cell. Economics of the cell, and when should they bother to transcribe and translate proteins, and when should they not? And depending on whether it's a biosynthetic anabolic process versus a catabolic process, is going to make the difference there. So this figure comes out of your book, and I like it, and it anthropomorphizes things and calls it decisions, but they put that in quotes, so it's A. But it's a good way to think about when a cell should turn off and turn on. And so let's just step through this. So remember we said you have catabolic processes and anabolic processes, biosynthesis and anabolism, same basic thing. Okay, so in the catabolic process, let's start over here. Actually, let's start with anabolic, it's a little easier to think about. Okay, so for biosynthesis or anabolism, if you have an end product present, right? I'm at a factory, I make a bunch of cars, I've already got a million cars out on my lot, so I need to make one more. No. So we have plenty of end product present. I'm a cell, I need to make tryptophan, but I've got tons of tryptophan in my cytoplasm, I don't need to make any more. Plenty of end product present. Then yes, I do have plenty of it. Then you go all follow the little arrows. Then no, do not synthesize the enzymes. The gene or operon is off. Off means nothing is happening. This hangs people up every time. Off means that gene is turned off. It's not functioning, it's not transcribing, and ultimately later translated. So let's say we don't have any end product. I don't have any cars on my lot. I need to sell cars. I need to make tryptophan, whatever it is. Then no, I do not have enough of my end product, so that gene or operon is turned on, so that I can turn it on, make the enzymes necessary to make tryptophan. Alright, biosynthesis and anabolism, a little easier to understand. Okay, and a catabolic process. So the substrate is present. Either there's a protein I want to break down, and I need to make the enzyme that degrades amino acids, or I need to break down lactose, okay, whatever. So in a catabolic process, if the substrate of that pathway, substrate being the thing that wants to be broken down, lactose, if that is not present, okay, so substrate present, no, there's no lactose available, okay, then no, gene or operon is off. So why am I going to bother to make all the enzymes to break down lactose if I don't have lactose around anyway? Okay, so yes gets a little bit more tricky. So yes, there's a substrate present, okay? And this branching is going to come into play throughout this lecture as we step farther into this lecture. So in the simplest sense, yes, the preferred carbon source is available, okay? But the next question is, or yes, there's a substrate available, but the next question is there a preferred carbon source available? This is going to make sense. It's going to come true in a couple more slides. So remember I told you, I asked you the question of what's easier to degrade, lactose or glucose? And everyone, of course, comfortably say glucose. Glucose is a monomer, lactose is a polymer. Now you're going to have to spend more energy getting your energy and getting your calories from lactose than you are from glucose. So in this case, glucose might be a preferred energy source. So yes, we have lactose present, but let's say yes, we also have glucose present. Okay? So if that's the case, and we do have glucose present, then don't turn that operon on. Don't turn on the genes to degrade lactose if I've already got glucose. Right? But let's say no, there is no glucose available. Yes, we have lactose, but no, we don't have any glucose. Then yeah, turn on the operon to break down lactose. Sure, we would prefer to have glucose. We don't have it, so let's turn on the operon to break down lactose. So in that case, it would be on. You guys comfortable with this? A lot of nodding and a couple of sleeping. Assuming the sleepers are bored. So as I said, all of this takes place on an operon. These are genes that need to be regulated. There's good energetic reason for you to either transcribe or not these genes. So what we do is we build that into an operon. We didn't do anything. It comes on an operon. Alright? And operons frequently start out with a regulatory gene. And that regulatory gene could code for something like that repressor or that activator. Okay. This is an example of the lac operon that you're going to see in more detail in the next few slides. It has, right here, it's labeled as a cap site. It's absolutely correct. I'm going to tell you what cap is in just a second. I'll tell you what cap is in just a second. That's your activator site for positive regulation. And then downstream of all that, you have the genes that make the enzymes that break down lactose. Right, and the terminator at the end. So again, yeah, here it is again. So this is, we're about to talk about negative control of the lac operon. So this is in fact the lac operon. So here, remember again, you have a positive site, the activator site, which is the cap site. I'll think about that in just a second. You have a promoter region. You have an operator. Note that the operator is where the repressor binds. That's behind the promoter, where the activator is in front of it. And the activator is positive regulation, so it makes sense that it would be in front of it. The repressor is negative regulation, so it makes sense that it would be behind that promoter region where RNA polymerase wants to bind. Okay, so let's step through this with respect to the negative control. Okay, so the lac operon under negative control, it can be under negative and positive or The lac operon under negative control is controlled by a repressor. Okay. When you have lactose in the environment, a little bit of it gets degraded at first and it produces a byproduct called allolactose. So when lactose first gets degraded, just a tiny bit, it's going to produce something called allolactose. Alright? In this case, you'll see that in a second. When there is no allolactose present, alright, the repressor is bound to the operator site. This is negative regulation. The repressor binds to the activator. Negative control, bound to the operator, repressor. No transcription occurs because there's no lactose present. Alrighty. When you have a teeny bit of lactose present, when you have some lactose present, you end up with a teeny tiny bit of allolactose. Allolactose binds to the repressor and removes it from the operator site. Alright, and when you don't have the negative control of the repressor bound to the operator site, transcription can occur. Alright, RNA polymerase can come down and get on that promoter and transcribe all these nice genes to break down all that lactose. Alright, so what's happening there is when there's lactose in the environment, the cell will have just enough of the enzyme available to make those first steps of allolactose. And then allolactose is the trigger, the sign that attaches itself to the repressor and lets it know, hey, lactose is around. Lets it know, removes the repressor from the gene, and then it can make a lot more of the enzyme and integrate all of the lactose. And this is kind of a, maybe you can call it a priming. Alright, so this is negative control of the lac operon. The lac operon is at the same time under positive control. Let me tell you how. So the positive control happens with regard to CAP, catabolite activator protein. And CAP is used for positive regulation of a lot of different things, which makes sense when I tell you why. So CAP is going to work in response to the presence or absence of glucose. CAP becomes active when glucose levels are very, very low. And it's going to make sense when we get into how this is a positive regulator. CAP is inactive when glucose levels are very, very high. In other words, when you have plenty of glucose around, why bother metabolizing lactose? When there's plenty of glucose around, we don't want to positive regulate the lac operon and turn on all the genes to create lactose. So it's going to allow for the preferential use of glucose. So what this does, and you can apply it, in this case we're talking about it with respect to lactose, but it's going to apply to other things as well. If glucose is around, there's no reason for the cell to try to make all the enzymes necessary to break down something else. Let's just get the glucose. So let's see how that works in a nice open text. Okay, so catabolite activator protein, all catabolite operons have this, and it makes sense. And I was just kind of alluding to that. It's not just, in this case, we're going to talk about lactose, but a lot of them are going to have this. So that if glucose is the easiest thing to break down, easier than amino acid, easier than lactose, etc. So if you have that available, glucose is going to shut down any other catabolic operon. Okay, so in this case it's positive regulation. inversely proportional to glucose concentration. When glucose concentration is high, CAP is inactive, and vice versa. Okay, here's the prefix. All right, so here's where you see this combination of negative regulation and positive regulation. So right off the bat, let's go to that repressor. Just remind you what I showed you a couple of slides ago. Here you have a repressor, okay? In this case, lactose is present. So because lactose is present, you have a little bit of allolactose bound to that repressor, so that repressor is off of the operator site. And because it's off of the operator site, transcription is occurring. You can contrast that over here where lactose is not present, so the repressor is bound to the operator site, and no transcription is occurring. That's the negative control. Okay. All right, so let's go with the positive control. Remember I said CAP is an activator. It's in my words. Not a repressor. It's an inducer. CAP is an inducer, and it binds to an activator site. The CAP site is an activator site. All right, and remember I said that CAP is inversely related